Question number 121. Bacteria which are utilized to produce gobar gas are: first option aerobic and heterotrophic, second aerobic and chemotrophic, third anaerobic and chemotrophic, fourth anaerobic and heterotrophic. The bacteria which are utilized to produce gobar gas or the biogas are methanogens. And these methanogens, they are anaerobic and chemoautotrophic. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 122. Match the columns and choose the correct option. A. Helicase joins DNA fragments. B. DNA ligase, synthesis of RNA primer. C. Primase unwinds the DNA helix. D. Amino acyl tRNA synthetase, activation of amino acid. Helicase, it's an enzyme which is helpful during the DNA replication. It unwinds the DNA double helix. So A matches with 3. So A matches with third. Option number 4 is ruled out. B's DNA ligase joins the Okazaki fragments which are formed on the lagging strand during DNA replication. So DNA ligase joins the DNA fragments. B matches with first. Option number 1 is ruled out. C's primase is a modified RNA polymerase. It synthesizes RNA primers. So C matches with second. So option number 2 is also ruled out. D. Amino acyl tRNA synthetase. It's an enzyme which activates the amino acids during the protein synthesis or the translation. So D matches with fourth. By analyzing the options, you can say the correct option for the given question is option number 3. Let's move on to the next question. Question number 123. Griffith performed an experiment where mice were killed when injected with a mixture of heat-killed S strain and live R strain of Streptococcus. But mice were alive when these strains were injected separately. Mice were killed because: first option, proteins from heat-killed S strain made R strain virulent. Second, RNA from heat-killed S strain made R strain virulent. Third, both DNA and RNA from heat-killed strain transformed the R strain and made it pathogenic. Fourth, DNA from heat-killed S strain transformed the R strain and made it virulent. Griffith performed an experiment on Streptococcus or Diplococcus pneumonia. When mice were injected with a mixture of heat-killed S strain and live R strain, then what happened? The mice were killed. Why? It means the S strain is smooth and the R strain is rough. It is smooth due to presence of a capsule which makes the bacterium virulent. And these strains are avirulent strains. So in this experiment, what happened? The DNA from the heat-killed S strains moves to the live R strain. Means the R strain takes up the DNA from the heat-killed S strain. So the transformation occurs here. So the proteins from heat-killed S strain made R strain virulent? No. RNA from heat-killed S strain made R strain virulent? No. Both DNA and RNA? No. DNA from heat-killed S strain transformed the R strain and made it virulent. It is correct. So the correct option for the given question is option number 4. Let's move to the next question. Question number 124. During transcription, the binding of RNA polymerase to the DNA is the prerequisite for the process to start. In prokaryotes, this site A consists of 6 nucleotide bases, B is TATAAT. C is located 10 base pairs upstream from the start point. D is present in promoter region. First option, only A and B are correct. Second, only B and C are correct. Third, only C and D are correct. Fourth, all A, B, C and D are correct. So during transcription, the binding of RNA polymerase to the DNA takes place at the promoter region. So this region is TATAAT in case of prokaryotes. So it's a 6 nucleotide bases, a long sequence. So these are correct. And it is located 10 base pair upstream from the transcription start point. So this is also correct. And it is present within the promoter region. So it is also correct. So the correct option for the given question is option number 4. Let's move to the next question. Question number 125. Select the incorrect statement. First option, triticale was the first man-made cereal crop. Second, crop plants grown in monoculture are highly prone to pest. Third, a wheat variety hemgary is resistant against rust pathogens. Fourth, mutations can be induced by infrared radiations. You have to choose the incorrect statement out of the following 4 statements. Triticale was the first man-made cereal crop. It is true. Second, crop plants grown in monoculture means the culture in which only one type of crops are grown and they are highly prone to pests. It is also true. A wheat variety hemgary is resistant against rust pathogens. It is also correct. Fourth, mutations can be induced by infrared radiations. No, infrared radiations do not induce mutations. They are alpha, beta and gamma rays which can induce mutations, not the infrared radiations. So the correct option for the given question is option number 4.